Today I am only going to discuss the real and present danger of having human fetal manufactured vaccines that can trigger autoimmune reactions and gene mutations in our children that could lead to very serious diseases. This is a graph of receptors that are part of our immune system. They are called toll-like receptors. The ability of the immune system to recognize molecules that are shared by pathogens like bacteria or viruses is in part due to the presence of these toll-like receptors that are expressed on the membranes of leukocytes, including dendritic cells, macrophages, natural killer cells, cells of the adaptive immunity, T and B lymphocytes. So the toll-like receptors have been implicated in many human autoimmune states. And in particular, I'm going to focus on toll-like receptor 9. When this receptor is activated by what is called hypomethylated DNA, that activation is associated with diseases that include lupus, rheumatoid arthritis, Crohn's disease, multiple sclerosis, systemic scleroderma, psoriasis, inflammatory bowel disease, type 1 diabetes, Sjogren's syndrome, myositis, and I want to emphasize encephalitis, which is brain inflammation. So when viral or primitive DNA fragments bind to toll-like receptor 7 or 9, it activates both innate and adaptive immune responses that will then be looking to find the same or similar DNA anywhere else in the body. So TLR9 has evolved as a general innate immune sensor for viral DNA, bacterial DNA, fungal and protozoan DNA, and other DNA that is unmethylated in certain areas or primitive DNA. What other sources of hypomethylated DNA are there? I've mentioned viruses. Cancer cells can be a source of hypo or undermethylated DNA. The DNA in patients with autoimmune diseases is often hypo or undermethylated, and fetal DNA is primitive DNA, which is also hypomethylated. So what happens when this, I'm putting it the wrong way, when this hypomethylated DNA binds to the toll-like receptor, it's on dendritic cells and cells of the innate immune system, and that releases cytokines that sort of trigger a general immune response or revving of the immune system. B lymphocytes also have toll-like receptor 9, and when the toll-like receptor activates the B cells, antibodies are made, and then those antibodies move through the bloodstream looking for the same or similar DNA to eliminate it, to kill the virus. This receptor is actually how some vaccines work, like the chicken pox. What I want to show you is how important this receptor can be. This is actually back from 1998, and what happens as a baby grows, what happens during pregnancy is the placenta begins to break down, and fetal DNA from the degrading placenta enters the mother's bloodstream, and when it reaches levels of about 0.22 nanograms per mil, so later in labor, it activates the toll-like receptor 9, antibodies are made to the baby's DNA, and they then travel through the bloodstream. In this case, instead of killing a virus, you get your baby. Okay. So just to summarize, DNA viruses like chickenpox activate the toll-like receptor 9. That's how we get immunity when we're given the vaccine. Okay. And fetal DNA that enters the mother's bloodstream also activates this receptor and causes labor. Okay, so just a summary of the fetal DNA. It's hypomethylated. It's about 300 base pairs in fragment length. And this is plasma levels. When it gets above 0.46, that is 0.22 nanograms in whole blood, it causes labor, which is a very profound biologic immune response. We are born with more nerve cells than we have later in life. And between birth, actually about a few months of age, and 3 years of age, 90% of our nerve cells die. So this is just data from rats. The black circles are a number of nerve cells, and you can see that there's a very rapid die-off. And a 1-week-old rat corresponds to about a 5-month to a 1.5-year-old human baby. So this slide is from autopsy data from humans, and you can see this very rapid nerve die-off in the human that occurs from birth to 2 years of age. So this is important background. If a child has been exposed to human fetal DNA, they will have antibodies to primitive hypomethylated DNA. There are people with genetic signatures that make them have hypomethylated DNA. And if they have antibodies to hypomethylated fetal DNA present when they undergo this brain remodeling, they can have a massive autoimmune attack against their own brain. And so where would a child be exposed to human fetal DNA to make these antibodies? And you've already heard from the previous speakers that when vaccines are manufactured in human fetal cells, they are heavily contaminated with human fetal DNA fragments. Okay, this is data from my lab where we measured the amount and the size of the human fetal DNA. On average, the MMR2 contains about 200 nanograms. So previously recommendations were that the DNA be limited to 100 picograms, but those recommendations have been raised twice and are now at 10 nanograms. So I just want to emphasize that this vaccine has single-stranded and double-stranded DNA close to 200 nanograms, so 20 times over, and a perfect size to activate the toll-like receptor 9. This is data from Merck on the chickenpox vaccine, and they report that there are 2 micrograms of the human fetal DNA in each shot that we give our children. Okay, so just for some perspective, micrograms are 1,000 times higher than nanograms. This is actually from an Italian group who measured the levels in Priorix tetra, and there are 1.35 micrograms per vaccine. The next two slides are just typical height and weight slides for toddlers. And I used these tables to find out the typical weight of a child when they get their vaccines. And based on that, there are algorithms that are available on the computer that will tell you the child's blood volume. So on the bottom rows here, these are the levels of human fetal DNA that trigger labor in their mother, which is a very profound biologic response. And up here are the contaminating amounts in these 3 vaccines, and these are the levels in the blood of our children when they are typically vaccinated. So this is about a 6-month-old child. This is about a one-year-old child. And you can see that all of these levels are above the levels needed to trigger labor. The scientists and the doctors who push these vaccines claim that the human fetal contaminants, they acknowledge they are there, are harmless. But anyone who says that either does not know anything about immunology or they are not telling the truth. So I want to talk a little bit about the other known danger of these fetal contaminants, which is a danger that they would insert into the child's genome and cause mutations. And we know a lot about this from the field of gene therapy. So I'm going to just introduce you to what's called small fragment homologous recombination, which is how we do gene therapy. So for gene therapy, targeted DNA is synthesized to specifically correct a mutation that a child has. And small fragment homologous recombination does not occur cross species. So just for example, when they make vaccines in a chicken cell, the chicken DNA is never going to insert into your child's DNA. And the best DNA to insert by this homologous recombination is hypomethylated DNA. And the best DNA is between 50 and 1,000 base pairs in length. And stem cells readily undergo small fragment homologous recombination. And so then I just want to reiterate what the vaccine contaminant DNA is. So this is a gel that is just looking at how this works. And they're using placental DNA fragments. Okay, so this is a mutated gene. This is the correct gene. You can see when they added these placental DNA fragments that the gene has been corrected in some of the cells. Okay, so this gel just shows you again gene correction, and in this column they use the placental DNA, and they get these bands again. But if they use salmon primitive DNA, there's no correction. There's no bands. So we don't need to be afraid of cow DNA. We're never going to grow tails. Okay, so this is in a mouse, and they wanted to do this gene therapy in an animal, and all of the animals had this happen in their bone marrow. And the cells that were changed were hematopoietic stem cells, and it was at a rate of 0.43%, similar to the levels of autism in the developed world. Okay. So the problem with having mutated stem cells is that they can give rise to immune cells that will enter the brain and not function normally. So again, this is just the concentrations of fetal DNA in our children's blood on the top. And this is the concentration in the mouse experiment, 1.9 nanograms per mil. And you can see that in several cases, these levels from vaccines are well over this level. And any doctor or scientist who claims that the contaminants are not going to do anything either doesn't know anything about gene therapy or they're not telling the truth. Okay. So there's quite a bit of real life evidence of how dangerous these mutations are. This is from a clinical trial where they did gene therapy for boys who lacked an immune system. Okay, so they took the blood from the boys, they isolated the stem cells, and they did this small fragment recombination, and then they gave the stem cells back to the boys. And 9 of the boys were cured. Okay, so 9 out of 10 were cured. 4 developed acute lymphocytic leukemia and had to undergo chemotherapy and transplant, and 1 of them died. So they wanted to understand how this happened. So what I'm showing you on this slide is this is when the cancer was first detected, and I want to point out that was 68 months after they did the gene therapy. And in this child, it was 33 months. Okay, so the original insertion did not directly cause the cancer. Okay, so other scientists did more research, and what they found was that the insertion occurs in areas of the DNA that gives that stem cell a proliferation advantage. And it also causes other additional mutations that cause the cancer. Now, the insertion doesn't always cause cancer because only 4 of the boys develop cancer. So we could have a cell with an insertion and a survival advantage that would be abnormal and yet not cancerous. So the scientists in this trial concluded that the leukemogenesis was likely caused by other genetic abnormalities, and they highlighted the general dangers of gene therapy. So scientists admit that this DNA could insert into a child's cell, but they say it could only cause cancer, not one of the chronic diseases that are so epidemic like autism. And they say that because they cannot conceive that an abnormal stem cell could impact an entire brain. But the scientists who say that either don't know anything about gene therapy or they're not telling the truth. And in many cases, I think it's because they actually don't know very much. So what this study is showing is these are young boys with a genetic disease that's fatal. It's like multiple sclerosis. And the boys will be doing karate 1 month, and then the next month they're unable to walk, and it's fatal. So what this doctor did was they did gene therapy for these boys, and the disease is an abnormal brain immune system. So this is brain disease measured by MRI, and then this is 1 month after they were given their own stem cells that had been gene modified. And do you see all of the gray dots? There's no evidence of disease in those boys anymore, proving beyond a doubt that in less than a month, the entire immune system of the brain can be replaced. So I want to just remind people the levels of fetal DNA that we are injecting into our children. And we know beyond a doubt that these levels can and do insert into the genome of stem cells. And they do cause a profound autoimmune response, which triggers labor. So the dangers of these contaminants have never been studied. And anyone who says they have, and this is an issue that undoubtedly cries out for serious scientific study. And finally, I want to say that the only thing that we need to do to guarantee that our children will be the sickest generation, to guarantee that we are willing to force mandatory vaccination and permanent brain damage on at least one of every 100,000 children, the only thing we need to do is continue to be silent. And I'll stop there. Thank you.